Okay, part 4 in our video series of predicting genes in a genomic DNA sequence. In this video, we will learn how Glimmer and GeneMark work to detect coding potential in a phage genome. These programs train themselves to recognize sequence patterns consistent with protein coding genes. They then search for those sequence patterns in the phage genome as evidence of protein coding genes. The programs Pecan and DNA Master both perform auto-annotations of a FASTA file using Glimmer and GeneMark to predict protein coding genes and their most likely translational starts. The result is a list of protein coding genes and tRNA gene coordinates. I will describe how Glimmer works first and then explain how two different GeneMark programs vary slightly from the Glimmer program. Gene prediction programs use Markov models to detect protein coding sequences. To build their models of protein coding genes, they first have to train on data sets containing known or assumed protein coding ORFs. They are essentially learning what the sequence patterns are in protein coding genes. After the training phase, it will create a statistical model, which it then uses to predict genes across the entire phage genome. The following is more than you need to know about gene prediction models, but understanding the gist of how these programs work may be useful at times when interpreting the data they produce. Glimmer uses an interpolated Markov model, which essentially means that for a given DNA sequence analysis, the program models the probability of a certain base occurring given the sequence prior to that nucleotide. Basically, given this sequence context of CTCGA, what is the probability that a C will occur, or a G, a T, or an A? In order to predict which open reading frames have sequence patterns that are indicative of protein coding, the program needs to first learn what those sequence patterns are that are associated with protein coding. In other words, the program needs to train itself on protein coding sequences. Glimmer trains itself using the genome that is being analyzed for coding potential. But how does it know which ORFs most likely code for proteins? Using the genome itself as input, the program extracts all of the long ORFs, at least 500 base pairs, using the assumption that usually the longest ORFs in a genome code for proteins. These ORFs provide the training data for Glimmer. It then studies the sequence patterns in these ORF sequences. It does so by determining the frequency of certain k-mers in the ORF sequence. A k-mer is simply a sequence length where if k equals 2, then the sequence length is 2 bases. It will learn patterns of single base, 2 base, and 3 base sequences. The longer k-mers, where k equals 5 or 6, allow the program to learn patterns with more context. Glimmer will determine the frequency of each sequence option for varying k-mers. For example, it will begin by looking at single base k-mers, where k equals 1. It goes through the sequences and asks how many A's, how many T's, etc. It then goes through the training set again and looks at 2 base pair patterns. How many AAs, how many ATs, etc. I am showing only the 2 base pair sequences beginning with A, but it will proceed through all of the 2 base pair sequence combinations. It repeats this process for each k-mer size, and the data set begins to become very large. After the training phase, it begins to build the statistical model. For each k-mer, what is the probability that an A, T, C, or G will follow a particular k-mer sequence? Here again is the 5 base pair k-mer with the sequence CTCGA. This formula asks, what is the probability that this sequence is followed by a C? Then the probability that the sequence is followed by a G, an A, or T. It then applies this statistical model to the entire genome. In other words, it looks for the sequence patterns it learned in the training set across all 6 reading frames of the entire genome. Here are the myriad of open reading frames that exists in the genome of Clipper. And here are the genes predicted by Glimmer. The blue and red boxes represent the genes that Glimmer predicted among all of these open reading frames. The result is also a list of coordinates for genes in our annotation. The GeneMark algorithms are different, but they work in a similar manner. There is a heuristic model that works similarly to Glimmer in that it trains on the longest open reading frames of the genome being analyzed, and then it predicts coding potential across the entire genome. This program is called GeneMark S. Here is GeneMark S output for the first couple of thousand bases of the Clipper genome. There are 7 genes predicted in the 3 forward reading frames. There is some coding potential predicted in the reverse-oriented reading frames, but it isn't strong relative to the coding potential observed in the forward-oriented reading frames. Let's zoom into predicted gene number 3. The line with the up and down ticks represents the open reading frame, with upticks representing the potential start codons, and the single down tick representing the stop codon. The coding potential begins at the genome coordinates of around 450 base pairs and ends at around 720 base pairs. There are 3 start codons that include all of the coding potential. Note the black box aligned to the x-axis. It aligns to the genome coordinates on the x-axis that GeneMark has predicted as the beginning and end of the gene, and therefore GeneMark predicts that the second start is where translation will begin. There is also a program called genemark.hmm, which we refer to as host-trained GeneMark. In applying this program, we are assuming that bacteriophage have the same codon usage pattern as the bacterial host on which it was isolated. GeneMark HMM trains on the bacterial hosts, such as M. smegmatis or Gordonia, and then it looks for the coding potential patterns in the phage genome. The host-trained GeneMark output is similar to that of the GeneMark S for the phage genome Clipper. But note that for some genes, such as gene 5, the coding potential is slightly weaker. Why might that be? It's important to remember the assumptions that underlie the genemark.hmm analysis, which is that the codon usage and coding potential of M. smegmatis is the same as the phage genome you are analyzing. And not only that the codon usage across the phage genome is uniform. It's important to understand how the coding potential you are using to analyze your phage genome was created as you make decisions about what is and isn't a gene in your phage genome. The major reason why has to do with the idea that bacteriophage genomes are mosaic. Remember, we learned that the genes that make up the phage genome come from a pool of phage and bacterial genes and come together to make a functional phage genome. We use the analogy of tiles that can be used in different combinations to create mosaic images. I'm going to conclude this module with 2 questions for you to think about. If genes in a phage genome, such as genes 39 through 42 of Clipper, were acquired from a bacterial genome or unrelated phage genome, will those genes have the same GC content and codon usage patterns as the phage genome? And which of the programs, Glimmer, GeneMark S versus genemark.hmm, are best suited to detect this newly acquired gene as a gene, and why?